I work in a medical center in the Netherlands as a bioinformatician, and I have a small group that develops applications and services to try and improve translational research and clinical practice through developing bioinformatics applications. I have quite a few slides, so it might be quite a long presentation, but I'll try to keep to the time. So thank you very much again. I do apologize. My slides seem to not be moving now. Try again. Oh, there we go. Thank you very much. So I would like to give an outline of how we work as a pathology department, and of course in my group of clinical bioinformatics. I'd like to take you through what workflows and platform technologies mean in the medical research in our faculty. I would then take you on to an area that we've developed in our own group, which is it's called Phantom Pathology Artificial Intelligence Platform. And then at the last part, just to show some of the projects we work on for precision medicine. So our department is set into 4 major themes. We have, of course, on the top left-hand side, clinical pathology, which is a standard way of monitoring how patients react or are diagnosed. We then, on the right-hand side, we have molecular pathology. This is to do with sequence-based methods, immunohistochemistry, all of those technologies that help us to give an improved diagnosis and prognosis to patients. At the bottom right-hand side, we have disease models and functional imaging. This is our sort of basic research and translational research in the laboratory to try and understand what those molecular events cause in the patients and how we can try and treat them. And then the part that I'm involved with as well with my team are looking at how we apply artificial intelligence to integrate this type of work or to use content from each of these modalities to generate predictive models so that we can try and improve our diagnosis, prognosis, and predictive outcome for clinical decision support. So, of course, you're probably all familiar with this idea of cancer diagnosis, prognosis, and treatment selection. And from the top part, the top line, you can see between diagnosis, prognosis, and treatments, there are many different aspects of what we're trying to do. Identify, where the tumor is from, do molecular subtyping, do prognosis and survivability, and of course, looking at precision oncology. How can we be precisely treating that patient based on their genetic background? And to do this, in the bottom section, you can see we have various techniques: genomics, transcriptomics, histopathology. And so all of those are included in what we use to try and identify the best outcome for our patients. And the new modality at the bottom there in pink is what we call spatial transcriptomics. And I'll come on to that a bit later. So to go on to this concept that I have of workflow and platform technologies, I'm hoping that the next few slides will make it clear what I mean by this. So in any department or process, you have something called the experimental workflow. So here on the left-hand side, we have different data types. It can be human tissue, it can be cell culture, it can be in vitro assays, it can be non-human modalities such as mouse or rat, fish, or whichever animal. And then you go through a sample processing part. The next step is measuring that. And then you do a statistical and data analysis. And finally, you come up to a standardized report. That's how we provide information back to the clinical groups or also back to our researchers as well. In parallel, we have our bioinformatics workflow. So, of course, you can see data or tissue is processed, it's measured. And so that measurement part would be, in this case, RNA sequencing. But for us as a bioinformatics group, then we would take that sequencing result, which is a set of letters, A, G, T, C, and we do some pre-processing and quality control. We then take those results and generate counts per gene. How expressed is that? How many counts per gene? For example, BRAF, KRAS, EGFR. That's what we'd measure in this normalization phase. And of course, because we measure things slightly differently between individuals, we have to make sure it's normalized. Because if I do counts say 10 counts in one patient and 100 in another, and I try and look at the ratio of those counts, well, I actually maybe just been measuring the difference in the total counts, not in the true value of what's expressed. So then we do statistical methods, and now we use deep learning and machine learning AI methods to try and analyze those results. And the last part there I would say is what I call platform technologies. So this platform technology are newer techniques. On the left-hand side, we would take a slide, a histopathology section, as you can see represented by that grid, and then we would identify in each of those cells what is the transcript profile. So unlike taking the whole tissue and grinding that up and then measuring the transcripts, we would actually identify the transcripts that relate to a structure. And on the right-hand side of that picture, we have the single cell or single nucleus RNA. So here we're really looking at the expression or the levels of gene regulation within a single cell within that tissue. You can merge these together because spatial doesn't look at single cell. It looks at approximately 10 or 20 cells at a time and try to understand the true expression between these different cell types, between different tissue locations. To go back one step, just to come back to this idea of targeted therapy, we have a project called PanNGS, which is looking at next generation sequencing in patients with pancreatic ductal adenocarcinoma, so PDAC. The top left, we see this sort of transformation of the tissue where some of the genes start appearing. So KRAS, CDNK, 2A, TP53, and SMAD. These are regulators that we see occurring throughout that process. And what we have a panel of genes, so these therapeutically actionable targets that work on DNA damage repair, MAP kinases, cell signaling, and PI3 kinases. And what we're trying to identify in the population that these seem to occur more prevalent in 15 to 20% of the cancer patients, and even more prevalent in what they call young cancer patients, so those less than 60. And this has been demonstrated if we used, on the right-hand side, in an article in The Lancet 2020, that if we use actually targeted therapies, we increase the survival time of these patients. So in our own study, it's called the PanNGS trial, we're looking at 250 to 300 individuals younger than 60 years old. We take a tissue biopsy, we send it off for sequencing. In this case, we use a US company, which is looking at fusion gene detection. And then the bottom part of that picture, we're looking at the tumor only, either at the transcript level, the RNA, or the DNA. And we've developed our own pipeline where it says tumor only workflow to identify variants in the RNA-seq that are specific to driving that process of cancergenesis, and actually identify targets which we can focus on with drug therapies, existing drug therapies. This is an ongoing trial, and we will present that work probably at the end of this year, early next year. But within that trial as well, we're also interested in the immune phenotypes. So there are several software applications out there that identify deconvolution. And that is to say a group of RNA signatures. So you can see all of, on the left-hand side, the RNA signature for different cell types represented by those different colors. And then we have what's called a signature matrix. So each cell type has a particular set of genes that represent that. So when we do RNA sequencing, just take the whole tissue and sequence it at the transcript level, which is represented by the bulk tissue or the blood RNA, we can then just use this cybersort technique to disentangle which cell types are in there, which is represented by the cell proportions. And so we've done this in this project to try and identify which types of B and T cells and T regulator cells differ between the liver metastases and the primary tumors. And this is just a small representation shown on the right-hand side with box and whisker plots showing the relative proportions. So anything that's in the pink boxes represents the liver metastasis, and anything in the primary tumors are represented by the blue boxes. So you can see here that it's very clear that the various cell types, macrophages, T cells, are differing between these two states of the disease. We've taken this one step further. So this concept of using and disentangling the cell types from a mixture population of cell types in liver cancer. So we're looking here, there are many different facets of liver cancer: intertumor, interpatient, intramural heterogeneity, interpatient heterogeneity. There are many risk factors in this process, and there are many stages, just like it is with with pancreatic cancer. So what we did here is we took our same set of data, or the concept of gene expression data, from either the Geo Omnibus, from the TCGA, and the International Genome Consortium. We took many different samples of expressed cell types from here, either hepatocellular carcinoma or cholangiocarcinoma, or different types of tumors within that state, within that range. But we also used here different primary atlases. These are the reference types. So in one case, we either use the normal liver, we use the tumor microstroma, or we use the tumor microimmune cell types. So effectively, we try and identify these. We use cybersort again, which is the method to separate them, and we try to disentangle from those group sets which fractionation of cells are responsible for the progression of that disease in that particular cancer type. So in this project here, we just are looking at this table here about whether the different cell types and the expression of those correlates between the atlases. So if we look at the left-hand side, it says cross-study normal atlas. We're looking at the two columns, the two vertical columns. We're looking at whether the stroma or the immune response are picked up if we're looking at hepatocytes. So depending on which atlas we use, we actually identify similarities in the gene expression, depending on those atlases. So for example, the top one, we can see the atlases, the hepatocytes are picked up by any type of atlas, whereas the NK-like cells are not picked up by the stromal atlases, but they are picked up by the immune atlases, possibly because those cell types are not stored within those atlases, so we cannot detect them. So the choice of atlas is very important when you're trying to disentangle the cell types within a particular RNA sequencing experiment. In this slide here, it represents the outcome, so the estimated cell types just for hepatocytes in figure A, and figure B as well for hepatocytes, but using two different atlases. So the bottom row, the horizontal, represents the experiments. You can see the GSE numbers, which are the gene expression omnibus numbers, so the data sets. Anything in light green represents a distribution of normal tissue. Anything in red represents a distribution of hepatocellular carcinoma. So we can see that the estimated fraction of hepatocytes, as would be expected, is higher in normal liver compared with the hepatocellular carcinoma. So we've taken this slightly further and we've said, okay, based on the population of these cell types and the gene expression, so we can take bulk gene expression from a human liver cancer and we can identify the survival type. So here we looked at overall survival and disease-free survival. And of course, the higher the hepatocyte levels, so either in blue or in red lines, we actually see an improved survival type of that individual. So it's a way of monitoring the individual and eventually, of course, then trying to identify targeted therapies to improve how we monitor and treat patients with hepatocellular carcinoma or some form of liver disease that relates to this type of modulation. I'd like to move on now to the spatial aspect. So this is now looking at the concept of taking a slide and identifying regions on that slide. So effectively, just as we do in a normal histopathology clinic, we would take the slides and stain them. It's shown in figure 1. And then you actually extract the DNA or RNA from that region. So for 10 or 15 cell types, you can extract it. And you can see in section 2, where it says select region of interest, you identify a region which could represent either the epithelium or the fibroblast layer or the tumor microenvironment. You then extracted those regions in part 3. You dispense that into wells in part 4, and then you send it to a library for sequencing. So you can sequence thousands of regions on a slide and relate those regions back to that slide to identify which genes are expressed in which regions of tissue, whether it's endothelial, epithelial, desmoplastic regions as well. So if we take this one step further, we are studying pancreatic cancer in our group with our collaborators in the faculty in the hospital. And so again, we're interested in this interaction between the tumor microenvironment, macrophages, B cells, T cells, and all of the other cell types in this interaction in the disease profile. So in our recent work, we used digital spatial profiling. This is a technique made by the NanoString company. So then we identified tissues using just standard gene expression profiling. That's in the discovery phase. We then took our results from that, so a sort of bulk idea of what's going on in profiling. We did some dissociation and deconvolution to identify which cell types are there. And then in the validation phase 1, we actually look at long and short survival times, so tissue from those individuals who have long and short survival times, so that's greater than 1 year, and use digital spatial profiling to identify what cell types are in which particular regions. And again, of course, we validated these validation stage 2 using immunofluorescence histochemistry to identify what cell types are there. And the article is represented by Frontiers in Immunology there, 2023. This gives you a representation here of what you actually see. So A, B, and C are representations of the ducts within the pancreatic cancer cell types. So the ductal area is represented in green. And the image above shows you just a small representation of what type of B cell counts are varying between the long-term survivors and the short-term survivors. So we can see that in the short-term survivors, there are less B cell interactions occurring in this cell type. And it went further. We managed to identify many different facets of tumor interaction that my colleague there, Dr. Dana Mustafa, who's the lead on this project, was able to identify which cell types were prevalent in the long and short-term survivors and whether we could influence those in the future for future targeted therapy. So I've taken you through now what we see as sequence-based methods to identify directed targets, directed therapies, precision-like medicine. I've now taken you on to understanding the concept of using histopathology and spatial representation so we can see how these cells interact at the transcript level. And my next phase is just to add a component in here, which is quite important. There is a reproducibility crisis. And the problem we have is that people write lots of different code. It's stored in different places, and it's not easily reproducible. Ourselves and our own group have tried to represent and try to reproduce some experiments, and it's quite difficult to do this. So there's a concept called FAIR, findable, accessible, interoperable, and reusable. And we also have this concept on the right-hand side, which is what are the 4 main components of trying to make something reproducible. So we've got computational environment, the data, the code, and the presentation. And it wasn't me that generated this figure at the bottom, but I think it represents how we understand this. So we have the environment, it's like your kitchen. We have your data, which here is represented by, in this case, potatoes. And then the code is you generating the methods. And then, of course, the presentation, how do you present it back to people? These are all important concepts. And there are solutions for this, actually simple solutions that are publicly available. There's one is called Galaxy. Galaxy is an application that's been developed worldwide, and it's available for free of use across many different countries. I think in Indonesia as well, and other countries, it's fully publicly available. What is it? Well, Galaxy is actually an application that has 3 components. It has a set of tools, a workspace, and a history, so you can identify these. This is all built together, so the user just gets to see this interface. And it's open bioinformatics. It exists for multiple omics data analysis. It's scalable to other modalities beyond transcriptomics, and it allows you to secure reproducible workflows that you can share with your colleagues. And that Today, I cannot present the whole of this application, but to me, I think it's a very important application to follow up on after you finish this meeting. What's important? Well, as I've shown now, you can use transcriptomics, but now we have this concept of single transcriptomics. So identifying single cell expression values. So the whole of this tool set, which is represented by all of these simple icons here, they represent the whole process where you could take data and run it through a pipeline as a scientist or as a medical person without the capability of needing to know how to program these skills. And we've gone one step further. So you can see Galaxy in the center, and you can have reproducible workflows stored in a public location called a workflow hub. So all of the workflows that we generate in our projects are stored centrally. So then you can log into a Galaxy, you get my workflows, and you can run them at that central location where you can see it says analyze workflows. But there are also public repositories as well. So when we finish our research work, transcript profiling or our spatial profiling, we load them into a location. And of course, because we're from Europe, we put them into the European archives. And at least in this European Genome Archive, which you can request access to, and usually on the majority of cases, you can get access to those. You can access the data, and we've developed a tool to bring that data into Galaxy without you having to go through some complex processes. So there are at least 4,500 studies there, very big studies as well. And these are mirrored in alternative sites around the world. The Workflow Hub has at least 107 plus standardized workflows to do many different types of analysis. And of course, Galaxy itself has at least 150 tutorials that allow you to analyze your data in the web, fully secure, without the need to install all these tools on your own application server. So the last part of my talk, I'd like to go into Phantom, which is our application for developing artificial intelligence tools to monitor and to analyze various aspects in precision medicine. So we have two aspects to our pathology group. One is the diagnostic. So we have a workflow on the left-hand side where we take digital slides, we scan them, we do whole slide evaluation, and we generate reports. And we augment our standard processes using commercial applications. In this case, we've used Visiopharm, and we also use our own inbuilt applications. On the right-hand side of the slide, you can see where it says the research. We develop our own applications. So the pathologists from the diagnostic world work with us on developing new applications. These applications range from anything from cancer-based applications, non-cancer-based applications, and also pathogen detection applications. So the central location there, it describes how we use image-based processing. And on the far right-hand side, it's a sort of schematic showing that we use phenotype data, genomics, proteomics, metabolomics, any type of data. So AI is sort of agnostic to how we do this type of work. So one of the applications that we developed early on was looking at how to improve the diagnosis of transplant rejection. So here we're looking at the different types of rejection, require different treatment strategies. The state of the art is to use histopathological consensus identification just by looking at the biopsy and trying to identify it. We used at the bottom there process of trying to identify rejection, non-rejection, or whether it was antibody mediated or T-cell mediated rejection, using gene expression from standardized nanostring expression profiles. So the two figures at the top where it shows A and B, it just shows that we were very good in predicting outcome based on this. If we took a sample, punch biopsy, took the gene expression from that biopsy, we can actually predict whether the individual would come to a point whether they would reject or not reject within so many months of transplantation. So it's an idea to use this in the future to understand not only the mechanism, because we can see which genes are important, but also how this process occurs. A second model we developed, this is using multi-omics again, is to look at prognosis-related subtyping. Which individuals are potentially going to fail in their treatment early on. So this was developed by two people in my group. And so we developed from multiple data sets, that's the multiomics data here, from many different sites. And then we developed a deep learning model. We took pancreatic cancer tumors, and then we all understood how pancreatic cancer developed as a moderate disease or an aggressive disease. It's still an aggressive disease, but relatively aggressive to one another. And we ended up with a predictive model. So our predictive model allows us to take a small biopsy of that tissue and predict how long the patient will survive, and also what sort of treatments would be useful. So we're extending this model now into treatment-based therapies to try and understand how these treatment affect this subtype, and whether that subtype or that individual will actually have an extended lifespan, which we hope we can do. In a third model here, I'm looking at something called integrative multimodality analysis. This project started off as looking at different types, subtyping of lung cancer. So in this case, we're looking at non-small cell lung cancer. But on the right-hand side, what we're interested in not only within the non-small cell lung cancer, which types of tissue and structures. So for example, in those different colors, we have stroma, acinar, papillary, and another papillary type. You can see in these biopsies, they're spread all throughout the tissue. But if we get a structure like this, can we identify by segmenting out these regions and generating a classifier whether we can predict survival and outcome for these individuals, and thus improve the way that we give decision support back to our clinical groups? So this is quite a busy slide, but you can see what we start off with on the left-hand side as a development. We look at our biopsies in the sort of big gray area over here, and then we need to annotate them. We have to annotate these. So our pathologists have to spend a lot of time annotating, and then we identify which regions are important. And then we go through this whole process of training and prediction to understand how good our models is. And following that, we need to repeat this on data that's unseen, so that the data comes in, we annotate it, and then we say, how do we perform in predicting our models? And we can get pretty high validation scores. We get up to, sometimes 60 to 75%, which sounds poor, but it's actually very good, even compared with the pathologists. So this is a model we're developing. This will be out soon. You can scan the QR code, and hopefully your student Theodora there has put it so that you can get to this code. So all our code will be made public as well. Where do we go with these methodologies? Well, we come to personalized medicine. And one project, before I carry on with the more cancer-based, is because we were all involved with the COVID pandemic. We were involved now in a project called BiCovid, which is to try and make sure that we can get content, genomic sequences, drug mechanisms, data shared around the world. So in this case, we were involved with a European project to identify research centers, consortia, and get that data represented in the sort of red lines into centralized databases to make this research open. So what we've developed is we've used Galaxy workflows to try and improve how we generate content. But of course, it's very important for the next pandemic that we improve how we work together to try and standardize this, make it open, but also be aware that we want to make sure we protect individual rights, and make sure that we look after people so that they are not exposed in the fact that they possibly are COVID infected, and maybe people are not very supportive of them. So this is a very important project, and if you get a chance to look at it, I think it's an important thing for personalized treatment, global personalized treatment. I would like to share with you something on a study we're doing. It's called Apollo. We're looking at transplant research as well, and I'll come back to the cancer ones in a moment. But here we're looking at whether we can regenerate the kidney. So kidneys get transplanted, they are in cold thermic storage, and then they are transplanted. Erasmus and one of the medical surgeons there is working on what's called normothermic. So they keep it at 37 degrees using a solution. And the idea is then, can we monitor the outcome? We've got a trial to see whether normothermic or cold thermic temperature improves the transplant capabilities and the functionality of the organ once it's transplanted. So we were involved with one component of this, and one component of that is to actually monitor using a camera. So it's like a spectral camera. It fires a laser at the kidney, and it monitors the perfusion rates in there. So the pathologist and the surgeon can look at this and identify, well, is there a good blood flow? We've taken that a bit further and we've said, okay, let's monitor these pictures in real time and identify whether we can predict from those images whether the kidney, once it's transplanted, will function better. And this is part of a bigger picture. Just to say that we have predicted it now, and those results are coming out quite soon, that we can actually predict from the images of the kidney during perfusion whether the kidney will survive and whether the kidney is regenerated enough to have improved function. It's part of a bigger study, and in this case, we're looking at not only the laser speckle, which is that graft assessment tool represented by Objective 2, but we're also looking at all the pre-data and all the post-data to see how this functions and whether there are other aspects that we can add to our deep learning model to improve this outcome beyond that that quality that we've generated already. Finally, I'd like to switch back to some of the newer techniques that are coming out. This is a paper that represents similar work that we do. So in this case, we're looking at neoantigens, those antigens that formed during the cancer process. It's quite a busy slide, but if you start from the top left and sort of circle around, a set of samples are taken from the tissue, or is taken from the blood, and then we do data generation, which is sequencing. We look for cancer variants, either with the tumor DNA, the normal RNA and DNA, and then try to identify variants. We also look at T-cell receptor profiling. We're interested in the haplotyping, and then we piece all this together so we can identify at some point sequences which we think are clearly carcinogenic or generated by the cancer. And then we can generate vaccines to these or peptides, and we can induce a response in the individual to try and improve how we attack the cancer, so more targeted therapy. However, there are lots of variability here. It's not a simple process. So at the top box here, we see the mutations that occur within within individuals. And of course, these may be 1 or 2 or 3 cells within that tissue that start out like that, and they eventually grow to a large tumor. But you can see at the top, there's something like 10 to the 9 possibilities of mutations in peptides. If we come down to section B, of course, then not only once we generate this peptide, we have to look at the T-cell receptor repertoire, and we have to look at the MHC class 1 and class 2 repertoires. And of course, these have variability. So effectively, we might be able to predict bioinformatically what's going on, but we may not be able to sample that data clearly from the individual to make these peptides very specific. So this is a problem we face. It's a problem of specificity, sensitivity, but also our prediction techniques. If we try to predict which species of peptide are antigenic, we're not that good at the moment. It gets worse in this case, because of course, if you look at the left-hand side, you have this concept, and we all have, of this clonal immunogenetic population. So effectively, you can see in the middle, it says there's an effective T-cell response. But as we treat, of course, these subclones grow out. And so we end up with different clones. So we've been treating with a particular clonal variant, and then a new clone comes in. But the therapy we've treated in, as we move left to right, changes. So we include new mutations. Those mutations are forced by the therapy itself. And we end up with subclonal variant, which grows again, which is non-immunogenic. So the therapy will have to go again. So these are just some aspects of what we see are important in developing this technology, which is very important. And one of the students I have works with the clinical group here, and we've developed a sort of process now. And I think by the end of the year, we'll be ready to start working on this in a clinical aspect. A tissue biopsy is taken, blood samples taken. Day 1 and 2, we do DNA and RNA isolation. Day 3 to 9, we do whole genome and whole RNA sequencing. Day 10 to 12, we're hoping to do neoantigen prediction, and we can do neoantigen prediction now from DNA sequencing and RNA sequencing. So can many labs in the world. Day 13 to 28, the idea is then to develop these peptides, and hopefully within a month from taking the samples from the patient, we'll be able to do vaccine treatment for these individuals, or peptide treatment as well. So just the last two slides, I would just like to say that what we use our toolbox from, which is represented at the bottom, we develop many tools. Our framework of using these tools is open science. We gather tools from other institutions around the world and give them credit for when we use them. And we use them from, on the left-hand side, I haven't presented these papers, but you have them in my CV. We worked on oncolytic virus therapy. We're looking at dendritic cell therapy with our clinical teams. I presented something here on our transplant rejection. I presented a little bit, just very briefly, on our pathogen monitoring. We use standardized workflows. And what I didn't present here is AMR detection and reproducible detection of these AMR environments, which can be used anywhere worldwide using very simple technologies using Galaxy. And the last slide I would like to show is something that I think is coming up, even though this was presented early on in 2017, this concept of precision medicine and single cell sequencing. I mentioned earlier that we use bulk RNA. And so bulk RNA, we don't know which cell types are in that bulk. We can try and deconvolute them, but there's a certain amount of error. If we go to the future prospects, which is the future medicine, more personalized diagnosis, we can use scRNA, single cell. So effectively, we actually monitor in those cells what's there. We can get the direct proportion, and then we can target drugs to those cell types and hopefully improve the patient survival and reduce the toxicity to those individuals. So I'd just like to show the support that I've received throughout this project. I'd like to thank my team there, and I have many other collaborators in Erasmus, which I didn't put on this slide, but I really think my students and staff who work for me have done a great job in helping the faculty and also medical research. And I'd like to thank you and take any questions.
Yeah, thank you very much, Professor Andrew. It's very, very interesting that the use of artificial intelligence now in the biomedical fields is really, really developed, especially for the cancer therapy.